So today we have Professor Hong Lee, who's Associate Professor at the University of South Australia, where he leads the Statistical Genetics Group at the Australian Centre for Precision Health. And I got in contact with Professor Lee after reading one of his articles in the Journal of the American Heart Association on genetic and non-genetic components that were modulated by lifestyle and environmental effects for cardiovascular disease. So today he's going to speak to a title that's very similar to his paper, but I think he appended the phrase that these might also be useful for other disorders too. So without further ado, Hong, please go ahead. And thanks very much for coming to present today.
Thanks, Andrew. Thanks for giving opportunity to explain our work and your interest. So I'd like to talk about the whole genome approach, discover novel genetic and non-genetic variance components modulated by lifestyle for cardiovascular health. But first part, I'll discuss about the cardiovascular health, but later also I will talk about the possibility can be applied to disease and disorder, binary tree. So I work with mostly with Swan in the picture, then a number of collaborators. So we start from the question that to what extent lifestyle factor modify cardiovascular traits. So as you know, the cardiovascular traits have both genetic and non-genetic components, and is well known that there is a genotype by environmental interaction. But here we say genotype covariate interaction because we call covariate because sometimes, for example, BMI, genetic effect of BMI modulated by smoking, then smoking is environmental, but also it has a genetic and non-genetic effect. So it itself has a kind of genetic traits. So we call covariate, so genotype covariate interaction here, but you can treat this as a genotype environmental interaction. Then now also we introduce some new concept about residual covariate interaction. So this is a residual heterogeneity, but I will talk about later in more detail about this one. And why? So because individualized lifestyle intervention is needed because it often one fits all approach doesn't work because there is individual genetic difference and individual environmental difference. So we use the multivariate reaction norm model to tackle this problem. So last year we introduced the novel multivariate reaction norm model. Then can you see my cursor? So hopefully that you can see. So in the right corner, upper corner, so this is a concept of reaction norm. So that is if two different genotypes responded differently when environmental is changed. So at that time, the most existing method only considering interaction, that is the genetic effect of, for example, BMI is modulated by smoking, therefore BMI phenotype is changed. So that is the interaction path. But in addition to this interaction path, there are a number of latent mechanisms. So not only interactions, but the same set of SNPs or kind of the genes in LD has an pleiotropic effects on the smoking as well as the BMI. Also, there is unknown factors, we call unexplained residuals here, but that's unknown factors that is not captured in the model. The effects can have effect on the smoking as well as BMI. Also, the effects of the BMI can be modulated by smoking. So we use this multivariate reaction norm model, then parameterize this is the main phenotype as BMI. And this is the genetic effects of BMI. This is the main additive genetic effects. So this effect is well known for the polygenic risk score that you can think. And this alpha 1 is interaction effects that's modulated by the covariate C, for example, smoking. Then in residual effect, there is effects not captured by genetic effect in the model, also has an interaction effects that we call tau 1 here. And tau 0 is kind of residual and noisy. Also, not only main phenotypes, also the covariate, for example, smoking has genetic effects as well as the residual. So here, not only we model the main genetic effects for the main phenotype, also we considering the correlation between alpha 0 and beta, there's a genetic correlation, alpha 1 and beta, there's interaction, genetic interaction correlation with the genetic effects in the covariate, as well as their residual effect, residual correlation between main phenotypes and covariate. So we model this all pathway then to tackle the correctly estimate G by E, so GC interaction. So this is a visualized mechanism. So again, this is the main phenotypes, for example, BMI, and this is covariate, for example, smoking. Then this is the main genetic effects, alpha 0. So this is estimate effect is here normally distributed. This is like a polygenic risk score based on additive genetic effects. And now we interested in this alpha 1. This is a GC interaction or G by E interaction effect. Then same as in main genetic effect, also you can get polygenic risk score, but this is interaction based on interaction effects, right? Then for the residual part, also you can get the same interaction effect, but here there's residual by covariate interaction effect. So we are interested in this polygenic risk score based on interaction effects. Then you can classify the top groups, that is blue, and middle groups, and bottom groups. There is a red line. Then according to this polygenic risk score based on interaction effects, then you can plot their kind of predicted phenotypes based on model along with the covariate value. Then this top 20% group, when covariate value is increased, then their risk is increased. But their middle group, they are robust across different environmental value. In the bottom group, when covariate value increased, then their risk is decreased. So this clearly shows that each individual has a different implication in their risk. So this top 20% group should be advised indifferently from the bottom group. So residual is also same thing happen here. So we applied this model to 23 cardiovascular traits and 22 lifestyle, then total around 500 analysis data points. So here, so for example, we applied the main phenotypes, BMI, waist-hip ratio, and so on. Then this horizontal line, this is all 22 lifestyle, that is alcohol, smoking, physical activity, and some kind of dietary things. So we use the from the ARIC cohort. So ARIC is around 7,000 individuals. So it's not that powerful, but still I think is sufficient to detect interaction. So we tested the three different sensitivity analyses. Then we found the 34 the most robust signals, that is shown in the gold color, the yellow color. Then as you can see, each trace for the cardiovascular traits has at least more than one interaction effect. So it's pretty quite often found that probably we can say that the interaction effect is ubiquitous across cardiovascular traits. So based on this finding, we used the UK Biobank. Then UK Biobank, we selected the main phenotype matched with ARIC, and also lifestyle matched with the ARIC we used. So it's not that many, but still a handful number of data points we can validate compared to ARIC. So most interaction we found from ARIC data can be validated, replicated in the independent data set using UK Biobank, right? So this is a kind of example that how much variance explained by the interaction. This is a GC interaction, and this is RC interaction. This is ARIC data set. The sample size is not that many. There's only 7,000. That's why the interaction, the proportion explained by interaction compared to total phenotypic variance is pretty large. Is quite optimistic, but I should say that probably there is a winner's curse, so maybe overestimated. So this kind of interaction should be replicated in the further study, whether to get the more correct and unbiased estimation of this one. And this is based on alpha-1. There is a GC interaction, genetic effects modulated by the environmental or covariate. So, for example, this HDL cholesterol, so predicted value and plotted against physical activity, like environmental variable. So we classify the top 20% and middle and bottom 20%. According to the polygenic risk score based on interaction effect. Then, as you can see, the top 20%, the HDL cholesterol predicted value is increased along with the physical activity increased. But bottom 20%, the direction is the other way around. So it can be, I think, have important implication in clinical practice. So based on this model, when you advise the potential patient or people to intervene or prevent their disease or this kind of cholesterol, or then according to this kind of considering individual genetic individual difference, then you can advise more precisely. So this is a kind of have some important implication precision health or medicine. So also other traces a similar example, whether when you classify according to polygenic risk score based on interaction effects, then each different group has different directions. So this is the same figure, but more kind of systematic. So this is one, for example, this is also based on the interaction effect, the polygenic risk score based on interaction effect. So the top 5%, and this is between 90 to 95%, and this is more row-length individual, and this is group. So we classify the first top 4 groups, then to see how much that one standard unit for the physical activity increased, then how much increased in terms of predicted HDL cholesterol. So it shows that increase around 0.3 when one standard unit is changed in physical activity. So this is previously I explained for the GC interaction, but now this is a residual covariate interaction. So compared to GC interaction, it looks the power is a bit low, but it gives a pretty similar pattern that when you classify it according to their interaction effect, then top group and bottom group, they have a kind of different direction. And also when one standard unit of a cigarette is changed, then they changed it around 0.99 unit of predicted white blood cell count. So this is an example how much environmental is changed, then how is effect on the predicted main phenotypes. Right. So this is more elaboration of the RC interaction. So because this residual covariate interaction is quite a new term, so I should explain more about this. So this is not in the paper, but this is the new study at the moment we are working on. So So RC intaction is residual, although we call residual covariate intaction or residual environmental intaction. So residual is kind of switched with the genetics. But you can think the intaction is not only genetic, but intaction is not captured by the genetic component in the model. That may include first the scale effects, that's heterozygosity, maybe phenotypic heterozygosity or residual heterozygosity. This is not really useful in the clinical care, but also you can think this another effect that is not captured by the GRM, for example, transcriptome or epigenomic or exposomic effects that are modulated by the covariate. Because this transcriptome epigenomic exposome is not captured by the genetics. So in our model, we capture genetic using the genomic relationship matrix. So this kind of different omics or different kind of effect can be captured, fully captured by the genomic relationship matrix. So the remaining factor not captured in the model can be partitioned as residuals. That's kind of noisy, but the intaction can be detected because of this kind of effect. So we tested the exposomic effects. The exposomic, I don't know if you're familiar with this term or not, but exposomic is kind of a totality of exposures. So all the considering exposures, then using that information. So we use around 30 different lifestyle and diet and physical activity exposures, then estimate the, like the genomic relation matrix, we estimate environmental relationship metrics based on this exposomic effect. So we call E here. So that is exposomic effect, like a genomic relationship metrics, but not genomics. This is based on environmental relationship metrics. So we pitted it in the same model, in the multivariate reaction norm model. Then to check it out, these exposomic effects are modulated by the covariate. So as you can see, we tested the 9 different traits: BMI, education, blood intelligence, and height, and so on. Then we tested the exposomic effect for these traits is modulated by this covariate, for example, age and principal components and some alcohol, smoking, and some physical activity. So this red font, this is p-value. I think this is p-value. So this is a significant effect. That means the exposomic effects are significantly modulated by this covariate. So, for example, smoking. So BMI, for the BMI, the exposomic effects of the BMI is significantly modulated by smoking for the BMI, education, and waist and weight, something like that. So this shows that the previously, what I showed in the previous slide, that we didn't consider this exposomic effect, but we estimate the residual by covariate interaction. Now we additionally fit this exposomic effect in the model, then find a significant effect. So this, the residual covariate interaction found in the previous slide may include this exposomic by covariate interaction. So hopefully this can spot the RC interaction is not only artifact effects, but this is partly genuine effect that include some genuine effect like exposomic effect. At the moment, we only considering exposomic effect in the new study, but we didn't consider transcriptomic or epigenetics yet. But if we can include this kind of different omics, for example, transcriptomic or epigenomic or other omics, then probably the phenotypic explained by this additional effect can be increased. So now, this slide shows that when there is a significant interactions, then if you don't consider interaction properly, ignoring interaction, then SNP heritability can be underestimated. So this is a ARIC data set, and this is UK biobank data. Then this gray dot is genetic variance, and this reddish red dots are residual variance. So as you can see, ARIC is a bit noisy in terms of genetic variance, and UK variant genetic variance is well lined up with this line. Sorry, I should explain what is the x-axis. So x-axis is G-REML estimate, that is a purely additive model, minus RNM estimate. So RNM is infection model. So purely additive model estimation minus infection model divided by G-REML estimation. So you can see, so when this is deviated from 0, that means the difference between G-REML and the reaction norm model. So, so that means genetic variance is close to 0. That means genetic variance estimate from G-REML and genetic variance from RNM is no difference. But residual variance, for example, for BMI, the residual variance from G-REML is almost 10% higher than the infection model. That means if you ignore the significant infection effect, then residual variance from additive model can be inflated like this. So around 10% is inflated. That means because SNP heritability is genetic variance divided by genetic variance plus residual variance. Therefore, if residual variance is inflated, that means SNP heritability is underestimated. So this shows that SNP heritability can be underestimated when you don't properly consider infection effect if in the presence of significant infection effects. So this is a summary of this paper. So we applied novel whole-genome approach, then reveals that lifestyle factors can modulate genetic and non-genetic effects on cardiovascular traits. Then these lifestyle changes are relevant to cardiovascular disease prevention. But as you can see in the slide, the associated cardiovascular health benefits are great for some individuals than others because of individual difference in terms of genetic as well as environmental. Therefore, we need the personalized lifestyle interventions. So far, what I show is mostly quantitative traits, but the question, because I know you are mostly interested in disease and disorders, so can this method can be applied to disease or disorder? So this is a preliminary result that currently we are working on. So we named as G by sum. So that is a genotype by environmental interaction model based on summary statistic like LDSC. So hopefully this can be appear in bioarchive soon. Also we can submit in a journal. So this G by sum is a LDSC version of mRNM, multivariate reaction norm model, or multivariate reaction norm model version of LDSC. Because so far LDSC is limited to additive model only. Please correct me if I'm wrong, but so far what I know is LDSC model is additive model that doesn't really consider the interaction effect. And mRNM, the problem is computationally demanding and need to assess individual genotypes. Also, it requires fitting genomic relationship metrics. Therefore, it takes a long time and large memory. So, but G by sum, at the moment, we currently working on overcomes the limitation of both approach. So this is a G by sum background. So this is a standard error, like an indicator of precision. And we tested, for example, 50,000 individual, 100,000 individual, and this is the maximum number of UK variant after QC. Then this is comparing standard error from the reaction norm model and G by sum. So given the same sample size, G by sum has around 1.5-fold higher. That means less precision compared to reaction norm model. But the advantage of G by sum, because of that, can be based on summary statistics. So it can use larger number of sample size. So for example, when GBISM can use 288,000 individuals, the full UK viable sample, actually reaction norm model is not feasible to apply this kind of sample size. So in that case, GBISM can use this full resource, but assuming that the reaction norm model is only use 50,000, then the standard error is reduced for GBISM almost 0.2, around 5 times higher precision. So that is ideal. Then when sample size increase, the GBISM, because that based on summary statistics like LDLC, the computational demand is not really increased. But reaction norm model is well known because the reaction norm model is kind of a GRAM approach. Therefore, the computational time is substantially increased. So when using more than 50,000, the difference is more than 1,000 times. So we take this advantage then to test all the binary trace. So here, disease and disorder is populations, not case control. So I should note that we haven't tested the case control, but only tested in the population. That is kind of UK biobank. So here, if there is no G by E and R by E, so we slightly change terms. There are two different studies. So previously we called GC interaction and RC interaction. Now we call G by E and R by E. Please don't confuse, sorry about this. So when there is no G by E and also there is no residual by environmental interaction, then when population prevalence is kind of only 2.5% to the 50%, the Taiwan error rate is pretty controlled. So I should note that we tested around 500 replications. So according to the binomial distribution that allow between 0.03 to 0.07 to control Taiwan error rate. So given this value, we should say that Taiwan error rate is pretty controlled. Also, there is no G by, and we introduced some kind of R by E. So in this G by sum, we only interested in the estimating G by E, then R by E as compounders. In that case, still Taiwan error is controlled, Taiwan error in detecting the G by E. Then the average of this all different simulation is close to 0.05 as expected. Yeah, and this case, now we tested with the case of population prevalence 10%, then introduce all different the confounders, for example, GE correlation and EE correlation. So GE correlation is a correlation between, actually genetic correlation between the main phenotype and covariate or environmental. So that's non-negligible. And EE correlation is a residual correlation between main phenotype and covariate or environmental. So there is a non-zero residual correlation between main phenotypes and covariate. And in this case, there are the GE and RE correlation both exist. And in this case, also we introduce, in addition to this GE EE correlation, then if we introduce some kind of R by E interaction, then what happened? Then, so this is to detect G by E. Then in the case, Taiwan error rate is always controlled. So every is close to 0.05. So. Yeah, and this one, so recently, I think several papers is concerned about collider bias because UK biobank, the response rate is very low. Therefore, the representativeness of the population is a concern. So in that case, we introduced some kind of moderate to severe collider bias, then to see whether Taiwan error rate is still controlled under this kind of all different confounders. Then we still find that the Taiwan error rate is controlled. So far I show that Taiwan error rate is controlled under many different scenarios for the binary trace disease or disorder in population. But this is this one I like to show that now is estimations. So as you know that when you apply binary trace, then estimate heritability as well as estimated interaction effects are all on the observed scale because your phenotype is 0 or 1, affected or non-affected. Then in that case, you should transform estimate on the observed scale to that on the liability scale. That is widely used in SNP heritability. So in that case, whether we tested whether G by estimate is working when we transform from observed scale to the liability scale, right? So this first panel explain when extreme case, population prevalence only 2.5%, either 90%, then they are mostly unbiased. The G by estimation is unbiased. We introduced 5%. So 5% by single covariate, the interaction is pretty extreme. Usually this is less than 1%. I'll show the real data analysis result later. But because 2.5%, then the number of cases is very small. That's why the power is reduced. Therefore, the standard error is pretty large. So in that case, we also tested the, this one is when we use 0.1, then varying the actual, proportion of G by E over the phenotypic variance. So when G by effect is around 2% and 1%, 2.5%, 5%, and 10% of phenotypic variance, then whether they are biased or not, we check it out after transforming. That is pretty nicely lined up with expectation, so it's not biased. But when population prevalence is extreme, like 2.5%, then when increasing the effect G by effect size, then it tends to be biased. So when you analyzing data using binary trace, then you should be carefully interpret your result, especially when prevalence is very low and effect size of G by is pretty large. And in this case, also there is some large compounders like G correlation, RE correlation. Also, the estimation can be biased. So this kind of bias is because when interaction is large, then the transformation may not work very well because the transformation always assume normal distribution. But when there is a large interaction, then the normality assumption can be violated. Okay. Yeah, so this one is from the G by sum model. We check it out whether SNP heritability, that is estimated mean genetic variance, is biased or not. So as you can see, the SNP heritability estimation is pretty unbiased, whether using extreme population prevalence or not. Okay. So this one, we check it out when there is collider bias introduced. So collider bias, the effect is a function of odds ratio. I won't explain about much detail about collider bias, how we simulate it, but you can think when there is odds ratio 1 and 1, there is no collider bias. There is 1.2, then there is some collider bias. So the kind of severity of collider bias indicated of the estimated main genetic variance. So this is when there is no collider bias, then we simulate the heritability around 0.5, then the estimation is close to true values. So that's mostly unbiased. But when there is collider bias, when we model odds ratio 1 is 1.5 and 2, that's a pretty moderate to severe collider bias. Then the estimated the main genetic variance, that's the SNP heritability, is substantially decreased. But in real situation, you don't often observe this kind of substantial reduction. So that means our correlated bias simulation is pretty severe. So even that severe correlated bias simulation, we show that the type 1 error rate is still controlled. So this case is we introduce another confounders like a different additional alpha interaction. Still, the SNP heritability is substantially underestimated under correlated bias. But if there is no correlated bias, then estimation is not biased. You give unbiased estimation. So this is a kind of indicator of correlated bias. So in real situation, when you using some sample that's suspicious of some selection bias, then if you estimate heritability is substantially lower than expected, then probably you think maybe that is because of correlated bias or some selection bias. So this is a real study application. So the it's pretty well known that there is a causal effect of obesity on hypertension from the Mendelian randomization studies from the several papers. So we applied the G by sum to check if causal relationship between obese variable like BMI and waist heat ratio and body fat percentage. Then between this obese variable and hypertension is because of G by E or not. So although the causal relationship between obese variable and hypertension is reported, but it's not clear that it's because of G by E or something else, right? So we applied this G by sum to this model. Then we applied the full UK sample after QC, that's around 280,000 individuals. Then we found that the genetic effects of hypertension is significantly modulated by BMI, given that p-value after Pomperoni corrections. So this confirmed that the causal relationship between obesity and hypertension reported from MR study is partly due to G by E. Okay, so in the kind of summary that multivariate reaction norm model or G by sum can be used for binary trace in random population sample, for example, UK Biobank. And type 1 error rate is controlled in general and robust to moderate to severe correlated bias, for example, UK Biobank. But we should note that we didn't consider case control data yet, which should be tested in further study. So we thank Swan and Jisoo and other collaborators from the University of South Australia and University of Queensland and University of New England. Also, we thank to ARIG and UK Biobank and funding bodies. And for software, for multivariate reaction norm model, you can download from this site. Also, G by sum will hopefully appear soon in the same webpage. Okay.
Thank you very much, Hong. That's a really terrific talk. Thanks very much indeed. Can I ask everybody now to type any questions they have into the Q&A box, or to use the chat box, or to raise their hand, and I will bring you into the conversation in a moment, if you wish. I wanted to ask just a quick question while you're getting started, which was, at the beginning of your talk, you presented two tables of GC and RC interactions, significant interactions. And I was looking down the list quickly just to see if they both picked out the same lifestyle modifier and exposure. Did you see a pattern amongst your results? Did the RC and GC analyses tend to identify the same set of exposures, or did they tend to differ? If they showed a significant effect in one, did they tend not to show a significant effect in the other?
So, sorry, you mean this slide?
I think it's further on than that. It was a table of results. Yeah. So here, do you tend to see the same trait and lifestyle factors appear in both of these analyses, or do they tend to be different?
That's different. So this one, GC interaction, the most large effect is HDL cholesterol, and physical PA2 is physical activity, and this is white blood cell count, and this is smoking. Then let's see whether same. Yeah, so this GC interaction to BMI is saturated fat. They are some similar, but I don't think there is a clear pattern. That's pretty looks independent, but I should check. But there is no. I don't think there is any correlation. So why you ask? Because you have some hypothesis or?
No, not really. I just wondered if things that tended to show genetic genetic interactions of lifestyle factors on particular traits tended also to show environmental effects also. But I don't really have a hypothesis. I was just curious.
Okay.
I'm going to just going to allow Mark Adams to speak now briefly. He has a question to ask.
Yep.
Mark, I think that should be you able to speak.
Hi, Andrew, can you hear me?
Yes, if you speak up a little bit more, that'd be great.
Oh, yeah, sorry, I just didn't have my headphones plugged in. Yeah, I'm just wondering about the potential to apply it to repeated measures data. So if we have, in some of our samples, multiple assessments of a phenotype across time.
Yeah, so this is a reactional model, but there is also well-established model about the random regression model. There is, I think, well suitable for the repeated measures. So I think that can be used, but that's not actually mRNM. That's a different model. But still, I think you can pick the repeated measures. Yeah.
Okay, thanks. So Alexandra Gillett, do you want to give your question?
Yeah, sure. Can you hear me?
Yes, loud and clear.
Wonderful. I was just wondering if using HapMap3 SNPs was recommended for creating the GRMs, and also should the genotypes be provided as dosages or made into sort of plink files and hard calls when creating the matrices?
Sorry, I missed your later part. So HapMap3 should be recommended. Is not that different, but what is your the later part? Sorry.
I was just wondering, when you're creating the matrices, should the genotypes be provided as dosages or hard calls?
So GRM, actually genomic relationship metrics, we used Plink or GCTA, so that can be used Plink file or dosage, right? I don't know. Yeah, I never use dosage. We usually use Plink file. So
wonderful. Okay.
Okay.
I'll use Plink.
So I've got a question here from anonymous attendee. Could you explain a little more about how you model the exposome in your exposome times covariate analyses? What factors are being considered in the exposome, and how are you differentiating the exposome from the overall residuals?
Yeah, so this is now a study we are working on, but I thought this would be well RC interaction, because RC interaction is kind of unknown factors not captured in the model, but I thought it's more explainable when we show that the EC interaction, that exposomic covariate interaction, that is a part of RC interaction. So here in exposomic, we construct environmental relationship metrics. So based on around 30 different environmental variable, exposure variable, including diet, physical activity, and so on, then you can column standardize. It's the same principle as the genomic relationship metrics. So in 30 variable, like m by m, now m is the number of environmental variable, then you can column standardize, then get the product metrics. That is exactly the same as the genomic relationship metrics. Then genomic relationship metrics is based on genotype, you get the similarity between individuals, right? But when you use exposure variable, environmental variable, then based on this environmental variable, you can find out who are more similar in their environmental and who are more dissimilar in their environmental. So you can get the environmental kind of environmental variance. That is a kind of C square. That is the same as heritability concept, but that is due to the environmental. Does it make sense?
I think so. Does I don't know if an anonymous attendee wants to say any more, but we'll move on to the next question if that's okay. Okay, so the next question is from Carmel Choi. Carmel, you're patched in.
Oh, hi. This was briefly mentioned, but I was just wondering if you could talk a little bit more about considerations for statistical power and sample size using this mRNA approach.
Yeah, because mRNA is a GRAMR-based approach, therefore it takes a long time, computationally demanding. So we comparing, and so maybe I can share. Let me, whether I can share on. Let me check it out. Yeah, sorry, I will show the paper. Sorry. Hold on. Sorry. So can you see the table? It's from the table 8.
Yes.
Is that okay?
Yes.
So here, when you around 5,000, then so RNM is kind of reaction norm model, and this is G by sum that's shown in the figure. So around 50,000, we didn't go further 50,000. So when you're using 50,000, then that takes around 1,800 minutes. So that's around it's pretty long time. But this is only using single CPU. So when you use a parallel, maybe depending on the server, you can use a 50 CPU or 100 CPU. Then it will probably reduce time substantially, but still it takes a long time actually. So at least maybe for the 50,000, probably few hours, even you use 100 CPU or something.
Okay, thanks. And we've got another question now from Chris Haley. Chris, that's you. That's you. In. I think you're muted at the moment.
Can you hear me?
Yes, yes. Quite faint, but yes.
Okay. How far can you push the GE sum approach in terms of breaking down the covariate or environmental effects into multiple effects in the same analysis?
Sorry, I don't understand. So break it down of the G by E from the R by E, or sorry?
Well, to actually break down the environmental effects and their interactions into the same analysis into the multiple effects of different potential environmental interaction. So that you can, for example, break the exposome down into effects which are more important and less important in terms of the interactions with genetics.
Okay, so one main difference between the mRNM and the G by sum is mRNM can model both G by E and R by E or E by E, but G by sum actually can't estimate R by E, it can only estimate G by E. So I don't know you're familiar with LDSC or not, but when you use LDSC, then you can estimate main genetic effects, but other compound is partitioned as interceptor. So when you applying G by sum, you only estimate G by E, but other R by E or other compounding is partitioned as in the estimate as intercept. So that's the main difference between G by sum and mRNN approach. So when using G by sum, actually you can't estimate R by E, so only G by E.
So if you went back to the mRNN model, you could break it down, but it would be computationally very demanding, presumably.
Yes, that's true. That's true. But our algorithm using when increasing multiple random effect, that doesn't really matter, because we use the average information algorithm. The AI algorithm has two kind of the model. One is a mixed model equation, as you may well know about, and what we use just working on the V matrix directly. So when using mixed model equation, then when random effects, number of random effects increase, then the computing time is exponentially increased. But when you're working on the V matrix, because they're just additive, there is no such increase. So the computational time is not much different. So it increased, but not that huge.
Okay, so have you tried breaking down these effects into multiple environmental effects then in the mRNN model?
Yeah, so we did. I think in the paper we mentioned using multiple covariate. I think more than 4 or 5 different multiple covariate is pitted simultaneously. I think when we explained the underestimate heritability, we did that. So 4 or 5 is pretty comfortably work, but more than maybe 10 or 20 is pretty challenging. So.
Yeah, I can imagine. Okay, that's good. Thanks a lot. Great talk, by the way. Thank you very much.
Thanks.
Thanks. I don't see any other questions. Does anybody want to have the final question before we sign up as we get closer to the half hour? No, I don't think there's anybody. So do you want to stop sharing your screen briefly, Hong Li, and we'll just say goodbye to everybody. So thanks very much for presenting to us today, Hong Li. This is a really excellent and illuminating talk, and I enjoyed it thoroughly. I should be going to read your paper again shortly after this talk. So thanks a lot. I dare say lots of people will start using this method in their analyses. Thanks to all the people who've come today as well, all the attendees, and thanks for all your questions. Hope to see you on the call again soon. Just to let you know that this has been recorded, and after checking with Hong Li, it'll be available afterwards, and I can send out a message to that effect. So thanks again. Cheerio, everyone.
Thank you very much, Andrew. Bye.
Bye-bye.